Bismillahirrahmanirrahim. This lesson is from unit number 17, Inheritance, and the sub-unit is DNA. And this DNA topic consists of many subtopics. For example, in this case, we are supposed to describe the structure of a DNA molecule along with subtitles. That is, a DNA molecule consists of two strands coiled together to form double helix. Each strand is made up of chain of nucleotides. Each nucleotide contains a base A, T, C, or G, and full names are not required at O-level syllabus. Bond between pairs of bases hold the two strands together. We will discuss what type of bond is there. The base pairs up in a specific way, that is, what is the pairing sequence. So this description fully covers what is required for O-level biology 5090. Only the structure of one DNA molecule, not its function or replication, is required in O-level syllabus. So the first subtopic is two strands coiled together to form double helix. So the DNA molecule is made up of two long strands and twist around each other like a spiral staircase, spiral stairs. So this coiled shape is called a double helix. The two strands run in opposite direction. They are anti-parallel. That is, if the arrangement of one DNA molecule, strand molecule is from 5 to 3 prime position, then the opposite strand, which is anti-parallel, should be from 3 to 5 from downward, from lower to upper direction. So the double helix gives the DNA molecule stability and compactness, allowing it to fit inside the cell nucleus. Then comes each strand is made up of a chain of nucleotides. So each strand is built from the repeating units, and the repeating units are called as nucleotide, just like the amino acid monomers and units in case of proteins. So therefore, DNA is described as a polymer, a large molecule made up of repeating units, just like the protein, which consists of amino acid polymers. The nucleotides are joined together in a long chain, forming the backbone of DNA strand. Then comes the subtopic. Each nucleotide contains a base A, T, C, or G. As we talk about the DNA, so there should be T. When we talk about RNA, so T is replaced by uracil, which is not the part of our syllabus in O-level. So every nucleotide has three parts: a sugar molecule, and in the case of DNA, the sugar is deoxyribose. That's why its name is deoxyribonucleic acid. Then the phosphate group, and third one is the nitrogenous base. A base consists of nitrogen atom in it, either A, which is adenine, T, thymine, C, cytosine, and G, guanine. The sugar and phosphate forms the backbone of the DNA strand because they remain same. The change is in nitrogenous bases. The base stick out from the backbone and responsible for base pairing between the two strands. So the bases of one strand, if they are arranging towards the right side, then the bases of the next strand, which is anti-parallel, they should come from on left side, so that they can come face to face and form hydrogen bonds, which is a weak bond, in order to stabilize the DNA molecule. So bonds between pairs of bases hold together the strand. The two strands are joined together by weak hydrogen bonds between these bonds are not covalent or ionic bonds, so they are weak hydrogen bonds and can be broken down by addition of energy. So these bonds form between specific base pairs on opposite strand. We have already discussed it. The hydrogen bonds allow the strand to separate easily during process like DNA replication and even the formation of RNA, which is the transcription. The bases always pair up in same way, that is A with T and C with G. So this is very significant and important because they are having much affinity with one another. So A with T will form two hydrogen bonds and C with G will form three hydrogen bonds. So a DNA molecule having more C and G proportion and ratio will be more stable. So this is known as complementary base pairing. So adenine always pairs with thymine using two hydrogen bonds. Cytosine always pairs with guanine because using three hydrogen bonds. So this pairing ensures the both strands are complementary to each other, and that is the genetic information is transported and transferred accurately and will be copied at the time of cell division. So by keeping the the DNA structure information at O level, this coming diagram is very informative and up to the level of O level. The structure of DNA molecule: two strands coiled together to form double helix, one strand, second strand. Then each strand is made up of chains of nucleotides. These are nucleotides. Then each nucleotide contains base A, T, C, or G. So this is a nitrogen base, and in this case, which we named it as the backbone of the DNA, consists of ribose, deoxyribose, and the phosphate group. The bonds between pairs of bases hold the strand together. So this type of bonding is hydrogen bonding. The bases always pair up, excuse me, in the same way, A with T and C with G. So AT and GC. So this is a sort of raw depiction of our level. So then comes the define a gene as a length of DNA that codes for a protein. Achha, let me to clear one thing. DNA, genes, these concepts are very much known to the students even at O level, but the requirement at O level is not beyond a certain limit. So we are supposed to limit ourselves within a particular range. So define a gene as a length of DNA that codes for a protein. The definition of gene is it's a length of DNA that codes for a specific protein. And even nowadays we name it as a polypeptide chain. What is the explanation? DNA is made up of many genes, each controlling a particular feature of organism. Each gene carries the genetic information that tells the cell how to make a specific protein. These proteins may be enzymes, structural proteins like collagen, fibrinogen, and functional proteins like hormones and other antibodies. The sequence of bases A, T, C, and G in a gene determine the order of amino acid in the protein. So this order gives the protein its shape and its function. So which then influence the characteristic of the organism. This topic, the detail of is in other topic. So in simple words, the gene is a section of DNA that gives instruction for making one particular protein, and this protein determines a specific trait, character, or function in a body. Then the third subtopic: explain the DNA the DNA controls cell function by controlling the production of proteins including enzymes. So let me to clear one thing at this level, that we should know that DNA is doing and producing nothing, just to produce, just to form proteins, and rest of the things, rest of the function of the cell, they will be performed by those proteins. So how DNA controls cell function? DNA acts as a genetic code for the cell. So this is important. DNA carries all the instruction needed for a cell how to work. So these instructions are written in the sequence of bases A, T, C, and G in the DNA and in a specific sequence and specific order. So each gene gives the code for making one protein. A gene is a section of DNA that provides the information for a specific protein. Different genes control the production of different proteins. Only some genes are switched on in each cell. So this is why, for example, muscle cell and nerve cells look and function differently, even though they have the same DNA as in other cells. So this thing is important, that some genes are being switched on in one cell and switch off in that particular cell, and this will make the fabric of the cell function. So proteins determine how the cell function. Most of the cell structure and activities depends on proteins. Some proteins are structural, forming parts of the cell like the cytoskeleton or membrane proteins, and many other proteins, structural proteins. Others are functional proteins, enzymes, hormone, transport proteins, antibodies, and others. So enzyme control all chemical reaction in the cell. So enzymes are proteins and they speed up chemical reaction in the cell. They act as biological catalyst. So every process such as respiration, photosynthesis, or protein synthesis, and each and every other chemical reaction is being controlled by the enzyme. So since DNA control which enzymes are made, it is therefore control all the chemical activities in the cell. So this diagram is showing the detail of this above mentioned topic. How DNA controls cell function. DNA acts as a genetic code for the cell. Each gene gives the code for making one protein. Proteins determine how the cell function. Enzyme control all the chemical function of the cell. So summary is that DNA controls cell function by controlling which proteins are made, and since proteins including enzymes carry out most of the cell structure and the reactions, the DNA indirectly control everything that the cell does. So the fourth subtopic is state that the sequence of bases in a gene determining the sequence of amino acid needed to make a specific protein. The knowledge of detail of nucleotide structure is not required. So what is the statement? The sequence of bases in a gene determine the sequence of amino acid needed to make a specific protein. For example, DNA contains bases arranged in a specific order. DNA is made up of 4 bases. This is just the repetition: A, T, C, and G. The order of these bases along gene is called base sequence. So when we go for DNA sequence nowadays, so the concept is the base sequence in that DNA molecule and the base sequence in a particular organism. The base sequence forms a kind of genetic code. So each group of bases gives the code for one amino acid. As we know, there are 3 bases required for one amino acid, and this will be the concept of A level. Proteins are made up of long chains of amino acid joined together. The DNA base sequence gives the instruction for the order in which amino acid must be joined. Different sequences of bases code for different amino acids and therefore for different proteins and even different functions of them. The order of amino acid determine the type of protein made. When amino acid join in a particular sequence, they fold in a specific shape. This shape gives the protein its function. Now, for example, an enzyme for an enzyme and even for hormone and structural proteins. So this is important because different amino acid, they will interact with one another to form a specific shape of that particular protein, and that particular shape of the protein will be responsible for its special function in the cell. If the base sequence changes, the protein may change. A change in the base sequence, that is mutation, which is not a normal thing, and mutations are the raw material for even inheritance, so which can lead to a different amino acid being added. So this is the concept of hemophilia and some other genetic disorders. This can alter the structure and function of the protein, sometime leading to genetic disorders. So what is the summary? The sequence of bases in DNA acts as a code that controls the sequence of amino acid in a protein. This determines protein is made and how the cell function. So base sequence in this DNA, A and T, just the representation that the bases are there. Otherwise, one 2 and 3, these are the strands showing the nucleotides. So sequence of bases and gene responsible for the amino acid sequence in a protein, and proteins when they get shape and they will be responsible for the cell function. So the last subunit understand that different sequence of amino acids give different shapes to protein molecules. Example explanation is the proteins are made up of amino acid. A protein is a long chain of amino acids joined together. There are about 20 different amino acids which can be arranged in many possible orders. The order of amino acid is determined by the gene, the DNA, because gene is just a sequence and segment of DNA. The sequence of bases in the gene decides the sequence of amino acid the protein, and even a small change in sequence of amino acid can alter the protein shape and function. Amino acid sequence affects protein foldings. Now this is important. After being made, the amino acid chain folds in a specific three-dimensional shape, because the sequence of the DNA or bases will determine the sequence of amino acid. Sequence of amino acid will determine the interaction between them, and the interaction will ultimately decide about the shape. So this folding is caused by the chemical attraction between different amino acids. The shape determines how the protein works. Different shapes mean different functions. For example, enzyme, they have active sites, the site where they can react with substrate, the molecule on which they are supposed to do their activity, whose shape fit a specific substrate. Structural proteins like collagen form strong fibrous shape, like keratin in nails, like others. Transport proteins have shapes them allow to carry molecules across membrane, for example, hemoglobin molecule. Therefore, changing the amino acid sequence changes the protein shape and may stop its function properly. Summary: the sequence of amino acid determines the shape of the protein molecule, and shape determines the function. Different amino acid sequence make proteins with three-dimensional different shapes and roles in the cell. So amino acid sequence, protein shape, and protein function, because if that protein is an enzyme, and this will be specific for its substrate. So this is all about the topic subtopic DNA. So Allah Hafiz from my side.